The wet tropics of far north Queensland harbors the planet's oldest remaining tropical rainforest and serves as a pinnacle of global diversity. Notably, the Atherton Tablelands encompasses the largest expanse of upland rainforest and serves as critical habitat for threatened and endemic species. Habitat loss, degradation, and fragmentation due to human activities such as land clearing for agriculture and urban development have led to a significant decline in remaining old-growth forests. Reforestation efforts have become an increasingly popular method to combat these threats and restore affected ecosystems.
So reforestation efforts on the Atherton Tableland aim to not only replant the vegetation cover, but also restore the intricate web of the ecological interactions and communities that sustain healthy rainforest ecosystems. By aiding the regrowth of native plant species, these reforestation efforts also promote the re-establishment of complex ecological processes and communities.
One way of examining the success of these restoration efforts is through assessing invertebrate populations. Invertebrate diversity and abundance are fundamental in assessing the progress of ecological restoration in reforested areas, as they serve as key indicators of ecosystem health. Invertebrates play a vital role in rainforest ecosystems, fulfilling diverse ecological roles such as decomposition, predation, and herbivory. By focusing on invertebrate communities, this study aims to provide valuable and new insights into the effectiveness of reforestation techniques and rebuilding ecological communities.
Our research takes place at Thiaki Creek. The Thiaki Creek Rainforest Restoration Project is a large-scale reforestation experiment which examines the most cost-effective ways to revegetate agricultural land in the Atherton Tablelands of North Queensland, Australia. Plots are planted with the same number of trees, but some plots are composed of 6 tree species and some composed of 24 tree species. We will be referring to these plots as low and high diversity treatments. At Thiaki, our project focuses on one aspect of revegetation success, being the re-establishment and composition of leaf litter invertebrate communities between low and high diversity replanting sites. We will collect leaf litter from sites planted with low diversity, sites planted with high diversity, and from remnant rainforest sites that haven't been cleared.
Now we're at our research site and it's time to collect some leaf litter. In each of our 50 by 50 meter plots, we collect leaf litter at the 10 meter, 20 meter, and 30 meter line. At each of those spots, we measure a 25 centimeter by 25 centimeter square on the ground and then grab all the leaves that fall in that spot and put them into a bag, making sure to leave nothing but bare soil behind. Now let's head back to the lab and show you what we do with these samples. All right, we just got back from Thiaki and it's time to get our invertebrates out of our leaf litter. So I've put some of our leaves under this lamp in this funnel here. What's going to happen here is the invertebrates really like it cool and damp, and this lamp is making it really warm and dry. So they're going to travel down this funnel into our collection pot. We've got an ethanol pot in there that they'll fall into, so we'll leave them in there for 60 hours, and then we'll take out that pot, bring it over to our microscope, and identify all our invertebrates.
We identified 596 invertebrates from 26 orders and grouped them into functional categories. Functional categories group invertebrates into the role they play in the environment. We found crustaceans, springtails, worms, and ants, which are all detritivores that eat and break down dead organic matter. We found predators and parasites such as wasps, centipedes, spiders, ticks, and mites, which prey on other living organisms, and herbivores such as thrips and leafhoppers, which feed on plant material.
Here we can see the difference in functional group proportions between plot types. There was a greater percentage of detritivores in low diversity plots and a greater percentage of herbivores in rainforest plots. The decrease in detritivores in high diversity and rainforest plots could be due to the fact that those plots have an established fungal community that is better at breaking down organic matter. We also looked at invertebrate abundance between plot types and found no significant differences. Finally, we found little to no difference in the environmental factors between plot types. Things like leaf litter density, soil density, and canopy cover, with the only exception being canopy height, which was highest in rainforest areas.
We found limited differences in invertebrate abundance between low diversity and high diversity plots. From an order level, leaf litter invertebrate perspective, focusing on low diversity plantings for revegetation could be more cost effective than high diversity plantings. However, ecological factors besides leaf litter invertebrates can have a huge effect on revegetation success and should be considered.
Our project examined invertebrates based on order, which is how invertebrate recruitment is usually assessed. However, this is still a very broad view and may be why we found limited differences between treatment types. The lack of difference in plots may be due to the difference being on a finer taxonomic scale. Therefore, future research may want to examine invertebrate communities based on genus or species. With limited environmental differences between plots, it's possible that as these plots age and become more similar in structure to the more mature plots they're trying to emulate, the difference in leaf litter invertebrate communities may only be visible through a more specific lens.
From an invertebrate perspective, our study sparks the idea that lower diversity plantings could be more cost-effective than high diversity plantings. If future research that looks at a finer taxonomic scale continues to support our findings, we could streamline operational costs and enhance conservation efforts in the wet tropics.